our Wnt drawing assignment. And so here's a description of it. Draw a picture of the Wnt signaling pathway based on this video, and take a picture and email this to me by Tuesday the 1st, anytime. And bring questions to class this week about the assignment, and next week you can even have a question in class on Tuesday and still get the assignment done later on Tuesday, or finalize your assignment. So used colored markers. You'll see it's going to be a very busy drawing, and it's a very important drawing. This is comparable to our clay model video that we started our course with. It'll be a complicated drawing. There are supplies outside my office, so set up there. You can come on over. You're welcome to come over with a classmate. And there's some tables nearby, tables out by the elevator, right near my office. You can move the supplies out there and the paper, and draw it there. And you can work with another student on the assignment. You're welcome to do that, but you have to make your own drawing and turn on your own drawing. If you have the supplies to do this at home, go for it. Don't buy anything, but you'll need some colored supplies, and you'll need to make it big. It has to be big because there's going to be a lot of information on it. You're going to draw and label more stuff than I have it. The drawing is based in part on figure 17. So this is a figure 17 assignment. You don't really need to look at that figure. I'll draw it here. It's about the Wnt signaling pathway. So there's Wnt written out there. Wnt is a protein. It's found in our body and every animal's body. And it's covered in chapter 17. And you can see it looks like a small part of chapter 17 because it is, but it's our example in the same way we talked about muscle differentiation and myodine, myogenin, and muscle gene expression. Wnt is going to be our example to be our general example that will talk about other signaling factors and signaling pathways using Wnt. And so here's that figure from chapter 17, 29, and it just shows that Wnt is going to bind, and it's going to stabilize a transcription factor called beta-catenin. So it's an example of regulating transcription like we've talked about, where we've talked already about how FOXO, a transcription factor, was regulated by a signaling pathway. We've talked about how ELK is regulated, and we've talked about how the signaling factor PDGF, platelet-derived growth factor, was released from platelets at the site of a wound and underneath the scab of every scab we ever have on our skin. PDGF is released, and it's stimulating the skin cells to grow and divide. And so we're talking about that type of an example, where in this case Wnt is the signaling molecule similar to PDGF, and beta-catenin is the transcription factor that's regulated in the process, similar to in the PDGF case ELK. Okay, and so here's an image at the start of chapter 17. There's the figure from chapter 17, and you can see that this is showing an image of hormones, and hormones are made by cells, and they're released into the blood, and they float around in our body until they bind to a receptor on a target cell. So that's a hormone. Hormones are called endocrine factors, and this is the same type of idea, but it's a local signaling factor called a paracrine factor, and paracrine factors don't go all the way throughout the body. They're local, just like PDGF at the site of a scab. It's only released at the site of the scab, so PDGF is a paracrine factor. And paracrine and endocrine are terms from the field of endocrinology. And so maybe at some really big universities in the United States, there are a few undergraduate courses taught in the topic of endocrinology. Definitely it's a big topic in medicine and medical school. So a person can go on and specialize in endocrinology and learn especially about hormones, but also these paracrine factors. And in this image here, this is Wnt. The purple molecule is Wnt. It's made by a cell and released and binds to receptor and affects that other cell. Okay, and so right now I'm going to just draw a simple outline, and so you shouldn't start drawing your final drawing now, but you can sketch this out because this provides a general image of what we're going to do. We're going to have a cell that makes Wnt, and it's called the source cell, and there are always many source cells, and that would make something like Wnt, and we just draw one. But understand this is cells in a tissue like that, and so Wnt is going to be made by this. So here's some Wnt protein being made. It's a protein, and it gets released from the cell and floats away. And in our example, we'll get to that, I guess, our specific example. But because it's a protein, Wnt must get made back here. So obviously there must be a Wnt gene, like that. So, okay, so the source cell is going to synthesize this signaling molecule, and then it's going to float to what's called a target cell. And there are always a bunch of target cells, and they have a receptor, and the receptors are always proteins, so a receptor protein. And they always have names, and ours happens to be a goofy name. This receptor is called frizzled, and it binds this. And when Wnt is bind, it doesn't come inside the target cell, it stays outside, but it activates a biochemical pathway that's called a signal transduction pathway. And we've already looked at those a little bit. We saw how a growth factor could stimulate a signal transduction pathway that involved RAS, RAF, MEC, and then phosphorylate the transcription factor ALK to activate it, stimulate growth. That's what happens when PDGF binds the PGF receptor in skin cells at the site of a wound. So the signal transduction pathway is going to ultimately lead to a cell response. The cell's going to do something. The target cells are going to do something. And often, they have new gene expression. And we like that. And so there's going to be, in the target cell, there's going to be some genes whose transcription was off, and now it gets activated. And that's going to happen because there will be a transcription factor that gets activated by that Wnt pathway, and our transcription factor is called beta-catenin. So there's a very general drawing, and your final drawing is going to look just like this, but it's going to be big, so I'm just showing you how big it's going to be, because we're going to label all this stuff over here, and we're going to label a lot of stuff over here, okay? So I say make it on at least 2 pieces of paper and tape them together, all right? So on we go. All right, so next, this is Wnt target genes. So Wnt can regulate a variety of different target genes. Wnt is a signaling factor that is outside the cell. Wnt is not a transcription factor, but we just saw how Wnt can regulate transcription factors like beta-catenin. And in doing so, Wnt can regulate gene expression. And so that's a really important concept. So far, we've talked about transcription factors directly regulating gene expression. Now we're talking about something outside of the cell, Wnt, that can regulate the activity of those transcription factors. So when we say Wnt has target genes, what we really mean is Wnt has target transcription factors that it switches on and off, and it switches beta-catenin on. And our example is going to be focused on a target gene that is called MYC. And MYC is a very famous oncogene. It was one of the oncogenes that was identified in those oncogenic retroviruses in the 1970s. And scientists know a lot about MYC. And MYC is mutated in a lot of cancers. And we're talking in the video today about normal development and normal stimulation of growth. But mutations can occur in genes for proteins on those pathways, lead to uncontrolled cell growth, like that. And you can see in this list in the columns, there are other target genes that are also involved in cancer pathways. And depending on the context, the setting, the timing, Wnt regulates a lot of different genes. We're just going to focus on one, but I just want to show you, here's a list of genes that Wnt regulates. by that pathway. So you can see that Wnt is at the top of like a big cascade of gene expression, and it can be different depending on the cells and the type of cells. Our example for our drawing is actually research that was done in the mouse during embryogenesis in the midbrain. So that's going to be our specific example. And Wnt is used over and over again in mouse development. And if it's used in mouse development, it's used in humans. So, we're talking about research in mice, but pretty much the same story would have happened in our bodies, in our embryogenesis, that Wnt helped regulate the brain and lots of different parts of the brain and lots of different parts of other parts of our body, like that. So a long list. Not only does Wnt regulate gene expression in humans and mice, but there are orthologs of Wnt in every animal. So here's just an image of that. Just talk for a minute about that animal in red called Hydra. And Hydra is the only animals that are more distantly related to us than Hydra are that gray blob at the bottom, sponges. Mammals are way up there on the upper left, and we are very, very distantly related to Hydra. There's a person that's looking at Hydra. Hydra live in the water, and they're really little. Down below is a green one because it picked up some photosynthetic algae, and it has a bud, so it's reproducing by its bud, and that Wnt signaling pathway is important in regulating the bud. Over on the left, Hydra have Wnt orthologs. That's why I'm showing you this. All animals do. If Hydra does and we do, everything in between does too. Salamanders and so forth. Okay? And you can see up there that it's not just Wnt. There's the word beta-catenin, and you'll see we'll learn the word TCF on this pathway as well. And so it's not just orthologs of Wnt, but orthologs of the Wnt signaling pathway. Hydra also obviously have an ortholog of the receptor frizzle. Okay? All right, so as I just mentioned, our example is going to focus on one specific example, Wnt signaling in the brain of a mouse embryo. Okay, and now we're going to draw it. And I'm not going to try to draw it slow and do a really good job and fill in everything. But you are. And so I'm not trying to get all the colors. I'm just going to try to draw it out and talk about it and be telling you where to fill in the gaps. Okay, so the first thing is to take like a whole side piece of paper and just make a big line on it like this, and that's going to be the cell membrane of our source cell, which again is going to be a bunch of cells that are all making Wnt. So this is the source cell. And it's going to make Wnt. And so there's going to be some Wnt protein that's going to be released from the source cell and float away. And so, again, since it has that, it must have, and over here you can make the nuclear membrane, so we're inside the nucleus because we're going to go find the DNA for Wnt, because Wnt's a protein. So Wnt protein has to be synthesized by the source cell, and so we got to find the Wnt gene. And here's the Wnt gene, and that's the transcription unit. And it has cis-regulatory elements, and so we know all about those. It has to have a promoter here, and then it has multiple enhancers. And I'm just going to draw 3, and they're different enhancers as part of the cis-regulatory elements for this gene. And one of them is actually way down here, because what we learned is the purpose of enhancers is they provide a binding site for transcription factors and thereby tether that transcription factor activity to the gene. And so it's like a leash, and so we can find an enhancer down here because it's tethering it to the gene. And it doesn't need to be upstream. We usually draw them upstream, but it can be downstream. And there's a transcription factor that binds to this downstream enhancer, and it activates Wnt gene in this specific example in the mouse midbrain embryo. And you don't need to learn the names of these transcription factors. It's just 3 of 2,000, and they're not as important as other names that we'll get to. But PAX2 activates it. And I'll just show you that in this example, there are transcription factors that are actually repressing expression. So we got a couple of powerful transcription factors upstream of the gene And both of them are working to repress the gene. And one of them is a transcription factor called 6-2, and the other is a transcription factor called EMX2. Oh, I should probably match that color up. So you know a lot about gene organization, and you can fill in these labels, like that's the transcription unit, the promoters, so enhancers like that. Label these up as much as you can. And then the PAX2 overcomes the repression, and so we get transcription, so you can show transcription occurring. And we got the primary transcript. It's going to get processed, and the mature mRNA will leave the nucleus, and so we got the mRNA out here. It's going to bind to a ribosome and get translated into Wnt. So that's the first part of our drawing, is that's what's happening in the source cell. And it's really important for us to appreciate that Wnt signaling is dependent on a source cell making it, and it's dependent on that transcription factor. And there are a variety of different enhancers and repressors that are upstream and downstream of Wnt genes. I should have said this before, but I didn't. In our bodies, we actually have 16 different Wnt genes, and our example is Wnt1. And we'll talk about that in class, because that's something we haven't talked about in our course, is that we have a family of genes that are all closely related, like cousins, and they're all called Wnt, and they're just numbered. And this is our specific example is Wnt1. So we got to get a cell to, a set of cells, a source cells, to make Wnt1, and there they are. They're making it. So you can see what's going on here is regulation of gene expression, in that pathway that's happening in the source cell to get it made and get it floating away. So I'm going to erase this, and I'm going to draw the Wnt binding to the target cell now. So on another piece of paper that you can tape together, I would say, because these need to be big, we'll put this here, and here's the cell membrane for the target cell. And it's a set of cells, and we've got Wnt out here floating around, and it's Wnt1 in this example, and it's going to bind to its receptor, and its receptor is called frizzled, and there's lots of frizzled on the target cell. Every cell's got to have the frizzled receptor in order to respond to Wnt, and some Wnt protein binds to frizzled and activates it, and that leads to a cascade of signal transduction. And I'm just going to draw over here in a little sort of side box that in this cell, in the absence of Wnt, beta-catenin protein gets degraded. So this is beta-catenin in the absence of Wnt, and it gets degraded. And it's getting degraded because another protein called GSK is a kinase that phosphorylates beta-catenin and targets it to get degraded. So this GSK leads to beta-catenin degradation, destruction in the absence of Wnt. When Wnt is active, we get the release of a protein called disheveled. And disheveled protein binds to GSK3 and blocks it from tagging beta-catenin. As a result, in the presence of Wnt, when disheveled is released and blocks GSK, we get stabilized beta-catenin. Like that. I'm just going to scroll back over here to show you that in our book. I'm just going to have to scroll to this image. There's the figure in our book. And on the left in our book, it shows in the absence of Wnt, the beta-catenin is getting degraded. And in the presence of Wnt, beta-catenin gets stabilized. And it shows it in more detail up there. And so this is a good figure in chapter 17 where it shows a lot of detail. So you see up in the upper right corner, there's disheveled, GSK, and APC, and check one, and accent. We're going to learn for our exam these words: went, frizzled, disheveled, GSK, beta-catenin. All those are going to be on our exam and are important words in this pathway. And that's going to be true throughout chapter 17, that we'll see quite a detailed signal transduction pathways, and we'll learn some specific factors along those pathways. Okay, so next thing in the target cell is, I'm not going to draw the nuclear membrane, but I'll draw our target gene. And so here's one of the target genes, and our target gene is MYC. And MYC has a transcription unit, and it has a promoter. And MYC actually has a whole bunch of different enhancers also. And but here's one, and this one has got a protein bound to it. And here's, I think, the one other name we have to learn, and that is there's a protein bound to it called TCF. And TCF is repressing MYC. And what beta-catenin does is it binds to CTCF and overcomes the repression and activates transcription of MYC. So we start getting RNA, and eventually MYC protein gets made. So MYC's a protein. And I'll just mention that MYC is a transcription factor. And remember, it's an oncogene, so when things get messed up with MYC, it leads to cancer. So MYC is going to regulate transcription of genes that regulate cell division. Back here, and we're almost done with this, back here we kind of come back here and just bring up another term. So beta-catenin is a transcription factor, but notice that it doesn't bind to the DNA, it binds to TCF. And there are transcription factors like that. Beta-catenin does not have a DNA binding domain, right? These kind of transcription factors are called co-regulators. So the ones we've talked about so far, like myogenin and MyoD and so forth, that bind directly to the DNA because they have a DNA binding domain, those are just called transcription factors. Beta-catenin is a transcription factor, but people tend not to call it that. They tend to call it a co-regulator. And co-regulators can be either coactivators, which is what beta-catenin is. And so instead of calling beta-catenin a transcription factor, lots of people just call it a coactivator, and it's understood that it's a transcription factor that doesn't bind directly to the DNA. And in other cases, co-regulators can be co-repressors. So we'll learn that term. And with that, I'd say all these terms on this slide are important terms. Every one of these terms is an important term for you to be able to talk about on our exams. And that's the Wnt signaling pathway. And like I said, in the first image in the source cell, there's some things like PAX2, EMX2, 6-2. Those are not ones that are the big showcase. This is the big showcase because it shows us how MYC is responding. Now, let's just remember that I said, it was gene expression in the source cell, right, that led to the production of Wnt, and then Wnt is affecting gene expression in the target cell. So this is a way for gene expression in some cells to affect gene expression in others. And what happens during our normal development is that cells talk to each other. They've got all the genes they need, and they talk to each other to make a plan to make all the different cells in the body. In this case, again, our specific example is Wnt signaling in the mouse midbrain in a mouse embryo. Okay, so we're good on that, and I just want to show you now the last couple slides, because you're done. That's the assignment. But look at this, because this is important, and that is we add beta-catenin to this list, right? Remember, it's a different kind of example. It's a dynamic transcription factor that leads to dynamic gene expression. MYC gets activated, MYC expression gets activated because beta-catenin gets stabilized. And so that's the example here. It's different than the previous examples, right? FOXO, ALK, estrogen receptor, heat shock transcription factor. those are switched on and off in different ways, each one of them. This one, it's stabilized, okay? Now, also notice that at the top I put this in red, one big picture. So for our course, and one big picture is this, and it's all about that clay model. The drawing you just did is a second big picture, and we have 3 big pictures, and here they are. First time you've seen this. And we'll talk about this in a class, of course, a lot. So our course's total big picture are these 3 things. First one, we've talked a lot about. What could talk about? Regulation of transcription, gene expression. The second one is chapter 17, regulation of cell signaling, especially signaling transduction pathways. Our model is the drawing you just did. We did it for Wnt, and we plugged in the Wnt story, but it's comparable for all the different examples in chapter 17 we'll talk about. And then our favorite cell response in response to cell signaling is going to be cell proliferation, growth, normal growth during normal development, or growing up, or puberty, right? We're talking about that example, adolescence, right? Like that, or wound healing, growth. And that involves regulation of the cell cycle. So that's our third piece. So we got these 3 big pieces: regulation of gene expression, regulation of cell signaling, cell communication, and regulation of growth, regulation of the cell cycle, okay? And why? Because mutations in those can lead to cancer, disruption of normal growth, and that's that. So we're going to stop here. Awesome. Thank you. And let me get out of here and stop the recording.